Hello everyone, my name is Palash Sashittal. I am a graduate student at University of Illinois at Urbana-Champaign, and I'm being advised by Professor Mohammad Al-Kabir. This work is in collaboration with Yunan Luo and Professor Jian Peng, also from UIUC. And today I'm going to talk about characterization of SARS-CoV-2 viral diversity within and between hosts. So as we all know, the SARS-CoV-2 genome is approximately 30,000 base pair long, and this figure shows position of known canonical ORFs in the genome. Now, typically for purposes of detection using RT-PCR, one would use primers that target specific regions in this genome. For instance, CDC recommends using these three primers for targeting three regions in the N gene. However, to study the evolution and transmission of this virus, one needs to sequence the whole genome of the virus. This is an example of the phylogeny and the transmission graph for the COVID-19 pandemic generated using NextStrain. Now, after sequencing, we obtain reads that are much shorter than the whole genome, and these reads first need to be aligned to the reference genome before we search for mutations, particularly single nucleotide variations with respect to the reference genome. So here is an example where 137 out of the 139 reads that map to the location 17373 have a variant allele. And this gives a variant allele frequency of 0.99. Short for variant allele frequency is VAF. And since only two reads match the reference genome, we could also attribute these to possible read errors. Now, mutations with VAF values that are this high are known as clonal mutations. If multiple strains of the virus are present in the sample, then such a mutation must be present in all these strains. On the other hand, for position 11335, we see a statistically significant number of reads that have a variant allele, and the resulting VAF is 0.2. Such a mutation must be present in a subset of strains that are present in the sample and can be seen as evidence of presence of multiple strains in the sample. So these kind of mutations are known as subclonal mutations. Now, many studies generate a consensus sequence for the samples where they would only consider mutations with VAF of more than 0.5, thereby ignoring most of the within host diversity. And these ignored strains can have implications in the disease outcome, and their identification can also lead to more accurate contact tracing. To uncover the underlying strains in the sample, one can perform deconvolution of the mixture to generate strains and their proportions for the given some number of reads. And we are interested in finding out how widespread is within host diversity in COVID-19 patients. And can we say anything about the individual viral strains that are present in these patients? So this slide shows a rough timeline of our analysis. So the first whole genome of SARS-CoV-2 was deposited to GISAID on January 5th of this year. We generate a discovery set of high quality bulk sequencing samples deposited to SRA before April 9th. This set is used to identify the number of viral strains and the mutations that are present in these strains. Our validation set comprises of 7,540 consensus sequences from GISAID and 416 bulk sequencing samples from SRA that were not included in the discovery set and deposited before April 22nd. And this validation set is used to validate the identified strains that we get from the discovery set. This is an outline of the subsequent slides of this talk. We first identify the number of samples that have subclonal mutations. We deconvolve these samples to uncover the underlying strains and their proportions. We construct the phylogeny on the inferred strains. Then we characterize the spatial and temporal distributions of the strains. We look at the mutations at the protein level and then identify samples with putative recombinant strains. So looking at the SRA bulk samples from both the discovery and validation set, we see that 62% of the total 621 samples have subclonal mutations. This bar plot shows the number of samples with different number of subclonal mutations. So clearly, within host diversity is present in most COVID-19 patients. Once we have identified the mutations and their variant allele frequencies, we can deconvolve the mixture samples to um, obtain the underlying strains and their proportions. So we take the bulk samples present in the discovery set and we formulate the deconvolution as a non-negative matrix factorization problem, which we solve using a penalty function based approach. Now for the 161 bulk samples in the discovery set, we identify 43 mutations that are shared across many of these samples, and we find 25 strains that can explain the observed variant allele frequency values. We can then use RAXML to construct a phylogeny on the 25 inferred strains that together have 43 mutations. We classify these strains into 4 distinct clades, and for the remainder of the talk, I will talk about the different properties of these clades that we were able to find. A few interesting observations in the phylogeny are as follows. So there is a single incidence of homoplasy shown with red bold letters here. Also, some of the identified clades and mutations have been covered in other publications. So, such as these two mutations here were used to characterize the so-called L strain in this National Science Review paper. Also, the mutation in the spike protein D614G, which has been linked to increased infectivity of the virus, is also present here in the branch going to clade 3. In this slide, we look at the geographical and temporal strain distribution using the GISID sequences in the validation set. The GISID sequences are annotated by a clade if they perfectly match to a strain or its ancestors belonging to the clade in the inferred phylogeny that I showed in the previous slide. So first we see that the phylogeny of our inferred strains and the identified clades agree very well with the phylogeny inferred using next strain. Next we look at the strain composition of the sequences um, from different countries. We see that countries like Australia have a diverse strain composition, while USA primarily contains strains that belong to clade 3. And finally, here we look at the temporal dynamics in this fish plot. On the x-axis we have time, and on the y-axis we have the proportion of samples that map to strains of a given clade. So we see that clade 3, shown in the orange here, is the latest to originate of all the strains, and even though it originates the latest, it eventually dominates over all the other clades in terms of proportion. The gray here is for representing GISID samples that did not map to any of the strains, any of the inferred strains. We also look at the strain-specific R0 plots in different countries using EpiSTIM. The details of this analysis are in our paper on bioRxiv. And we see that clade 3 has a high transmissibility compared to the other clades, shown here for Australia and USA. Next, we perform a protein level analysis of the mutations. Out of the 43 mutations, 28 were non-synonymous, including 27 missense mutations and 1 nonsense mutation. We were able to get solved structure for 3 of the proteins. from the protein data bank. For the rest of the proteins, we performed homology modeling using HHspread. Here we show the proteins for which we get a high confidence homologous template. The first observation we make is that 24 out of the 26 mutations are near or on the surface of these proteins, and these mutations are not deleterious to the protein structure and are likely to be transmitted because of their little impact on the protein structure. We look at the number and position of unique non-synonymous mutations in the GISID samples mapping to strains from different clades. We see that clade 1 and clade 2 have similar mutational loads, while clade 0 has a lower mutational load and clade 3 has a slightly higher mutational load. Moreover, the mutations in the samples mapping to clade 3 cover more locations in the genome, most noticeably spike protein and ORF3A. Finally, we categorize the bulk samples based on the strains and their proportions inferred by our deconvolution method. The first kind of sample is an ancestral mixture that has strains that lie on a single path from the root to the leaf in the phylogeny. So in this example, we have two strains where one strain is an ancestor of the other. A multiclade mixture, on the other hand, has strains that belong to different clades. So in this example, we have one uh, strain that belongs to clade 2 and another strain that belongs to clade 3 in this sample. Finally, some of the samples are homogeneous, that is, they have a single strain. One such sample is shown here. Now, for some samples, it is not possible to deconvolve the mixture with the inferred strains. This sample, for example, has mutations that belong to different clades, and you can get pairs of mutations such that the sum of their VAFs is more than one. When this happens, there exists at least one strain that has both those mutations. So here we show the overlap of mutations from different clades. So there is at least one strain in this sample that has both mutation 8782 and 25563, even though these mutations belong to different clades. So one possible explanation for this would be a recombinant strain originating from two strains, one that belongs to clade 1 and another that belongs to clade 3. We see that out of the 621 bulk samples that we analyzed, 41 samples show putative recombinant strains. The bar plot here shows the number of samples of the other categories. We have already seen homogeneous, ancestral, and multiclade samples. A cladistic sample is one that has strains that are not direct ancestors of each other, but belong to the same clade. In conclusion, we analyzed 621 bulk sequencing samples and 7,450 consensus sequences to characterize the viral diversity within and between SARS-CoV-2 hosts. We found evidence for within host diversity across phylogenetic clades, putative cases of recombination, multi-strain, and superinfections, and distinct strain profiles across geographical locations and time. For future work, we expect that our algorithm can be applied to analyze viral diversity at the organ level as well, further correlating the identified strains with outcomes such as severity, mortality, and morbidity can lead to some important insights. Finally, the inferred strains can also be used to improve the reconstruction of evolutionary history and also for more precise contact tracing. Finally, I would like to acknowledge the Al-Kabir group for feedback and discussions during this work. Also, this work was funded by these 3 grants by NSF. Thank you. I'll be happy to answer any questions.